For an epithelial originating tumour cell to become malignant, it must invade the surrounding tissue. This begins with one driving tip cell detaching from the primary tumour and leading the invasion. By down-regulating E-cadherin expression and increasing N-cadherin and vimentin expression, the cell-to-cell interactions between epithelial tumour cells decreases, allowing the cell to become more mobile. This is a process termed as epithelial mesenchymal transition. The second challenge to invasion is the basement membrane that confines the primary tumour. The expression of MT1-MMP on the surface of the tumour cell allows the excavation of the cell into the stroma through the direct cleavage of collagen 4 in the basement membrane. The third challenge to invasion is the presence of ECM proteins in the stroma. These involve collagens, proteoglycans, and other fibrous materials. One of the first proteins encountered by the cell is collagen 1. The tumour cells break through this layer by activating MMP2s. Tumour cells are also assisted by macrophages and fibroblasts, which also release MMPs, heprinases and serine proteases, further breaking down the surrounding ECM, allowing migration of the cell. The body has a mechanism to suppress MMP activity through the release of TIMPs from surrounding ECM cells. TIMPs bind the zinc binding site on MMPs, inhibiting MMP action. The degradation of the ECM also leads to the release of growth factors present in the ECM, which positively feed back to the tumour cell, enhancing growth and mobility. Hepatocyte growth factor is the predominant growth factor that promotes motility of epithelial cells. Meanwhile, the cell matrix interactions are modified to enhance motility. Integrins expressed on the surface of the tumour cell are altered, changing shape and orientation of the cells, allowing for greater motility through signalling cascades. A commonly expressed integrin in invasive tumours is AVB3. To move through the matrix while degrading it, the cytoskeleton of the tumour cell attaches its leading edge to the ECM via integrin interactions. It contracts at the trailing edge, propelling the cell forward. As this occurs, the cell continues to secrete proteases to degrade the ECM in front of the leading edge. For a single cell, this is termed mesenchymal migration. Now that the tumour cell has invaded the ECM, it has achieved one of the hallmarks of cancer and is termed malignant.